Well, welcome back to our Biology 180 lecture series. And so today we are going to be covering functional groups and polymers. And this fits in our last lecture. We covered a little bit about carbon and isomers. And now we're going to be taking those carbon structures that we talked about in this lecture and introducing the concept of functional groups in there and how we add chemicals to those carbon structures to make it more useful. Understanding functional groups and polymers is actually going to be critical because in our next 4 lectures, we're going to be talking about the major classes of biological molecules. And this is going to be carbohydrates, lipids, nucleic acids, and proteins. And we will see functional groups come up again and again as a topic as we discuss these various macromolecules. And then all of them have polymer forms. And so today is really going to give us a foundation for understanding the rest of the topics in this particular unit. So, what are functional groups? So functional groups are a collection of atoms. So they're a collection of atoms that can be attached to carbon skeletons. And then they change the properties or functions of that carbon skeleton. Properties and functions of that molecule. Now, if you happen to take organic chemistry, then you're going to learn a whole bunch of different functional groups. I think there's as many as 30 or 40 different recognized functional groups. But in Bio 180, we want you to know really 8 common functional groups. Functional groups in Bio 180. So these are the ones that you're going to see over and over again. And so we're going to present those functional groups. We're going to draw them. And we're going to use this table. So we're going to fill out this table right here with the various functional groups that we want you to know in Bio 180. And so we're going to start. And for each functional group, we need to know a couple things about it. This will help us understand material as we go through the course. We're going to look at, obviously, the structure of that functional group. We're going to talk about its polarity. This is a topic that we covered way back in Unit 1, talking about ionic or polar or nonpolar, and that has a big bearing on the characteristics and functions of the functional group. We'll talk about some of its properties that are unique to that functional group or particular to it. Some of the functional groups, if you have a molecule that has that particular functional group, then those are known by a particular name, or those compounds are known by a particular name. So we'll review that and give you some of the common examples where we'll see those functional groups as we go through the Bio 180 course. So the first functional group we're going to start with is going to be alkyl groups. So alkyl groups, this is kind of a common or more general name of it. Generally, when we talk about alkyl groups in Bio 180, we're going to see 2 other terms for them. We're going to see them called methyl. Methyl is a type of alkyl group. And then another one that's not uncommon is ethyl, with methyl groups having 1 carbon and ethyl groups having 2 carbons. So we'll draw a methyl group right here. And a methyl group is simply a hydrocarbon. An alkyl group is some kind of hydrocarbon chain. It just has carbons and hydrogens. This would be a methyl group. You'd have, right here, an R. So R is a common nomenclature that we use in chemistry. And when you see an R in a molecular structure like this, I tell students that it stands for the rest of the molecule, all that stuff that I didn't want to draw and don't care about. So in this case, you have some kind of R group. And then the methyl group itself would just be a carbon with 3 hydrogens attached to it. And again, a methyl group is a type of alkyl group. There are other types. An ethyl would have 2 carbons here. They could have a propyl, which would have 3 carbons. And these are some kind of attachment to the molecule. So that would be an alkyl or a methyl group. Now, as we learned earlier, the functional group itself is composed of entirely carbon-carbon or carbon-hydrogen bonds. And so this is going to be nonpolar. In fact, that is our only nonpolar functional group that we're going to learn about. And so its properties, well, it's hydrophobic. So things with alkyl groups become more hydrophobic. They are not very soluble. So they are less soluble in water. And generally, we find that alkyl groups are not really all that reactive. Okay? So less soluble in water, they're hydrophobic, less reactive. Compounds that have an alkyl group in them or methyl are often referred to as just hydrophobic compounds or alkyl. And some of the examples, where are we going to see this? We will see it in lipids. So for example, fatty acids. When we learn about fatty acids later on as we talk about lipids, we're going to see examples of alkyl groups. And then a majority of the amino acids that are used in proteins have alkyl groups attached to them of one form or another. All right. Our next functional group that we're going to talk about is going to be a hydroxyl group. Now, the structure of the hydroxyl group is going to be pretty much just what its name says it is. It's going to have a hydrogen and an oxygen in it. And so a lot of times you'll see it, here's our R group, and then it's just written up as an OH together, paired together like that. So we have a hydrogen right here and an oxygen right there. Now, because this oxygen is very electronegative, you can imagine that the bond between that oxygen and hydrogen is going to be polar. And so the polarity, we're just going to write polar there. And so properties, well, because this is polar, as you might imagine, it becomes very soluble in water. So now when we add hydroxyl groups to things, they do become soluble in water. In fact, I like to talk to students when I talk about this about syrup. So if you guys have ever had syrup, it's really thick. If you've ever made syrup before, the recipe ends up being about 2 cups of sugar to about 1 cup of water when you make that sugar, and that's because, or syrup. And that is because the sugar, there's actually more sugar in there than water, and that's because that sugar is so soluble. It really just soaks up that water. And so things that have and I should say that the sugar itself is filled with these hydroxyl groups, and that's what makes it so soluble. So when you add a hydroxyl group to it, it makes it soluble. We'll bring this term up later on, but it has that hydroxyl group has strong partial charges. And we're going to see our hydroxyl later on when we talk about polymers and chemical reactions. We're going to see that hydroxyls make, I'm going to write this up there, good attackers. And then I'll explain what I mean by good attackers a little bit later in the lesson. Compounds that have a lot of hydroxyl groups on them, common name is alcohols. So we call compounds with hydroxyl groups alcohols. We're going to see this in carbohydrates, like glucose. So carbohydrates. We're going to see a lot of hydroxyl groups. There's some really important amino acids. We're going to see it in a molecule called glycerol involved with lipids. So we really see hydroxyls all over as we look at different molecules. They're one of the most common functional group that we see. Moving on to our next functional group. After hydroxyls, we're going to put down a group called carbonyls. Okay? So carbonyls are going to be the first example. They have an R and a carbon. Okay. So this is the structure of what a carbonyl looks like. And really the functional group is this carbon that is double bonded to an oxygen. So we now have two bonds going to this oxygen. Now, because we have an oxygen-carbon bond there, and that's a polar bond, we do put this in the category of polar, so its polarity is polar. And we have added, so as far as properties go, it is soluble. And then we're also going to say that this has, in parentheses, an activated carbon. And what we mean by that, as we talk again about reactions later on, we're going to see that this oxygen, the two bonds, we've got 4 electrons in those two bonds, holding the carbon to the oxygen atom. But because oxygen is more electronegative, all 4 of those electrons are being pulled more to the oxygen. And those negatively charged electrons are going to make this oxygen have a partial negative charge, and this carbon have a partial positive charge. But it's actually a very strong partial positive charge because 4 electrons, not just the normal 2, are being pulled over that oxygen. That means that this has a very partial positive charge, and we're going to see later that this makes it a good target for attackers to come in and make a new bond. So by activated carbon, what we mean is that's a carbon that is going to be prone to either forming new bonds or breaking old bonds or both. And so that's what we mean by that activated carbon. So carbonyls, compounds that have carbonyls, we actually have 2 different names for them. They could be either ketones, or in some cases they're called aldehydes. Whether it's a ketone or an aldehyde depends upon the placement of this carbon in a chain. And there's 2 ways that this can work out. So I'll try and draw one. If we see the carbonyl group at the end of a chain, meaning that it's the last carbon here, so the final bond is a hydrogen instead of another carbon. If it's the terminal, we call this a terminal carbon. So if the carbonyl carbon is a terminal carbon, then this compound would be called an aldehyde because the carbonyl group is on the end of the chain or terminal. On the other hand, if we see the carbonyl somewhere in the middle so that the carbonyl carbon is connected on both of its remaining bonds to another carbon, it's somewhere in the middle of the chain, then this would be a ketone molecule. So we've got ketones and we've got aldehydes. Now that can be a little bit confusing, so a little trick, it's easy to get those 2 mixed up. A little trick that I use to remind me of this is aldehyde starts with an A, and A just happens to be on the end or at a terminal of the alphabet, right? And K, ketones, just happens to be the letter that's right in the middle of the alphabet. So I remember that ketones have the carbonyl in the middle, just like K is in the middle of the alphabet. And aldehydes have their carbonyl on the end, just like the letter A is on the end of the alphabet. And so that's the trick that I use to remember those. So ketones and aldehydes is the name of compounds that have carbonyl groups. Name of the group is carbonyl. Name of the compound is ketones and aldehydes. And students often, for some reason, mix that one up a lot. They don't tend to do it with the other compound names, but they'll often say, yeah, the ketone group. It's not a ketone group. The whole molecule is a ketone. The group is a carbonyl group. Where do we see carbonyl groups? We are going to see carbonyl groups in nucleotides and that's largely, and I guess we'll see them in carbohydrates also. All carbohydrates always have at least one carbonyl, well, have one and only one carbonyl group. Our next group is going to be a carboxyl group. Now, carboxyl group, the carboxyl group obviously sounds very similar to carbonyl group, but it's different. So it's got an X in there. And its structure is similar but different. So in a carboxyl group, we are going to have, here's the rest of the molecule, and we're going to have a carbon here. Now this carbon is bound to double bonded to one oxygen, and then single bonded to another oxygen. And so it actually has 1, 2, 3 bonds to oxygen. A lot of people you may have seen already, and this is the wrong way to see it, but sometimes it can be helpful. The carboxyl group looks like a combination. It looks like we took a carbonyl group right here and then mashed it together with a hydroxyl group. And structurally, that's kind of what we've done. However, the full group is actually the whole thing. And together, those parts are somewhat symbiotic in that the whole structure has new and different properties than either the hydroxyl or the carbonyl. And so you have to be able to recognize this as a carboxyl group and not just a carbonyl or hydroxyl put together. Now, so what are the properties? We're going to see same properties actually as carbonyls and alcohols. This carboxyl group, it's got those oxygens. It becomes very soluble. So it is soluble. It also, because again, we have a carbon, we have bonds being shared or electrons being shared with oxygens. Those electrons are drawn toward the oxygen. Really strong partial positive charge on that carbon. Only now it's even more so because we've got additional oxygens pulling those electrons away. So this is actually also has an activated carbon. It's even more activated than the carbonyl carbon. And we now add another property, and that the carboxyl groups are what we call acidic. So we didn't do the polarity yet. So we're going to call this ionic. This particular functional group is so polar that it actually crosses over the border and becomes an ionic functional group. What happens is this hydrogen right here is able to fall off of or leave the overall group. And so when it leaves, then the group gets a negative charge on it, becomes an anion. So this is, and we call that, it's giving up a proton, so that makes it acidic. So carboxyl groups are one of 2 acidic functional groups that we have. And we'll talk about the other acidic group next. Compounds that contain a carboxyl group are called carboxylic acids. Again, reminding us that they are acidic. And where do we see examples of this? We're going to see examples in, no surprise, fatty acids and amino acids. Our next functional group that we're going to put up here is going to be a phosphate group. On our table, phosphate groups are actually the hardest to draw. They are the most difficult because they have the most atoms and the most complex. But it's not, once you get used to it, it's not super hard. We're going to draw an R group. As the name implies, phosphate, you might think, oh, it's related to phosphorus, one of the elements, and it is. So we're going to draw a P right there for our phosphorus atom, and then we're going to draw oxygens on all 4 quadrants of that phosphate, and then one of those oxygens attaches to the R group, and then H's. And then we're going to add a double bond, actually, to one of them. We're going to remove that H and have a double bond. So the phosphorus has 5 bonds connected to it. One is double bonded to oxygen. The others are all single bonded to the oxygen. As you might guess, those oxygens all pulling on it. This is going to be ionic, so the phosphate group is an ionic polarity, and it is soluble. It now has an activated, it doesn't have an activated carbon, but it does have an activated phosphorus, for the same reason that we had an activated carbon before. All of these oxygens are drawing electrons away from that phosphorus, which gives it a big partial positive charge on there and will make it a good target for attack later on. We'll talk about that. And then it is also acidic. So that's our second acidic group. Carboxyl groups are acidic, and then phosphate groups are also acidic. Compounds that contain phosphates are just called phosphates or phosphoric acids. And where do we see examples of this? We're going to see examples in a group of molecules called phospholipids, which are, as you might guess, a type of lipid. And then we're also going to see them in nucleotides and nucleic acids. That's where we're going to find our phosphate groups. The next functional group we're going to deal with is actually a pretty common one. It's the amino functional group. So the amino group is going to have the rest of the molecule, and it has a nitrogen. And then that nitrogen just has some hydrogens on it. So it looks like that. That would be an amino group. It is actually going to be, as far as polarity goes, so we'll write up this is its structure. Polarity. Properties, compound names, and examples. The amino group is actually going to be ionic, and I'll explain that in just a second. And in fact, it's ionic, well, and it's basic. Meaning that it actually gains a hydrogen ion. Again, it has a lot of polar bonds in it, so it's very soluble in water. And it is actually, we'll call it a weak attacker. And I'll show you what I mean by that. One of the biggest properties that students also often confuse is basic, meaning that it acts as a base. It draws protons out of solution onto it and will result in a basic pH when you have an amino group on something. So students really have a hard time with that and often get it confused. So I'm emphasizing the fact that amino groups are basic. Compounds that have amino groups are called amines. And the most prominent example that we find are amino acids, obviously, in their name. They have amino in there. So we have amino acids. And then nucleotides also have a part on them called nitrogenous bases. And so the nitrogenous bases obviously have nitrogen or amine groups on them. Let's maybe for a second pause and looking at this amino group, let's explain. Again, you don't have to necessarily memorize this or even really know it. You really have to just know that amino groups are basic. But to help you better understand that, let's look at an amino group again and look at why it might be basic. So if we draw an amino group, here's the 3 bonds that it makes, and then we have an unbonded pair of electrons there on that amine. And to understand why this is a basic, we also have to draw a water molecule nearby. So remember in biology that this, whatever molecule it is going to be surrounded by water. And we learned earlier in an earlier lesson how water can dissociate and act on itself. And sometimes a water molecule, just as a quick review, we said that this hydrogen up here, it's got sharing electrons. The oxygen has the electrons most of the time. And this hydrogen starts to get a little bit jealous, would like a little bit more electron time, if you will. And so sometimes it will hop down onto that oxygen, and that's the dissociation of water. And then we get a hydroxide, negative charge over here, and a hydronium ion over here due to that dissociation of water. This doesn't do much to the pH because as soon as the hydrogen hops over, it's just as likely to hop back. In other words, hydrogen is trying to decide between an oxygen and an oxygen, and it really doesn't get any more electron time with either one. They're both the same. It's like going from one bad relationship to another bad relationship. Nothing changes. And so they just go back and forth. And it doesn't really affect what we call the pH. However, now consider if I have an amino group somewhere in there, this same water molecule, again, hydrogen facing a decision. Oxygen is hogging the electrons most of the time. It's more electronegative. And it sees a pair of electrons over here. And so it jumps ship, thinking, hey, maybe it's better in this relationship. Maybe I'll get a little bit more electron time. And so it hops over. However, in this case, it's true. Remember, nitrogen is not as electronegative as hydrogen is. And so this pair of electrons that gets shared actually does share more equally. The nitrogen does not hog it as much. And so now the hydrogen, rather than hopping back onto its original oxygen, stays there. Why? Because it gets more electron time. When it stays there, it adds an overall plus charge to that amino group, and it will lower the overall pH of the solution, so it becomes basic. So again, it's basic because hydrogens will actually leave water molecules and move toward the amino group because they get more electron time, because that nitrogen is less electronegative. And so that's why our amino groups are basic and have a positive charge on them, because they're actually drawing hydrogen ions out of water. So that's our amino groups. Our next group is going to be called a sulfhydryl group. And sulfhydryls are going to have, again, an R group, and then they're now a sulfur. So instead of taking an oxygen, we're now going to take a sulfur atom and connect it. And then it has just an H there. Sulfhydryl groups are weak polar, is their polarity. And their properties, again, they're weakly soluble. One that is often noted by students is that they are smelly. So Compounds that have sulfhydryl groups have a very strong smell to them. In fact, if you've ever gone to a natural hot springs and you walk in, that kind of sulfur smell that you get is because there's a lot of compounds that have sulfhydryls in them. And that's where that smell is. It happens a lot of people call it a rotten egg smell for the same reason. Rotten eggs develop a lot of sulfhydryl groups in them, and that's how they smell, why they smell like rotten eggs. So they smell it, rotten eggs, we'll put here in parentheses. Rotten eggs in hot springs. That's probably what we'll know. We'll talk maybe a little bit later. They come up mostly we'll see them actually only in one place, and that is in amino acids, at least in the molecules we deal with. And compounds that contain sulfhydryl groups are called thiols. And again, this is one that students often confuse. Thiols are compounds that contain it. This is not a thiol group, it's a sulfhydryl group. Compounds that contain it are thiols. Our last and final group is called amide. That's right. Our last and final group is called an amide group. Amides, remember over here we said that carboxyl groups looked kind of like a hydroxyl and a carbonyl had gotten smushed together. Our amide groups are kind of a smushed together group too. We are going to see the same carbon double bonded to oxygen, and then we're going to see it bound to a nitrogen instead of another oxygen. And so that's going to be our amide group and what its structure looks like. Again, it's very good to compare it, or it's easy to compare it to a carboxyl group. If you know how to draw a carboxyl group, you know how to draw an amide group. You just take this oxygen, erase it, and put a nitrogen in its place. Add another hydrogen to balance it. So that's what an amide group looks like. Amide groups are polar. Remember that the carboxyl groups were acidic, but amide groups, again with this nitrogen, you kind of have this combination of almost being acidic, but this one being basic, and so they balance each other out. That's not quite how it works, but it explains it. So these are not ionic, they're just polar. We do have very soluble, so the presence of both the oxygen and the nitrogen make those compounds very soluble. They are activated in the sense that we have an activated carbon like we do the others. It is not acidic, so I'm not going to put that down. Compounds that contain the amide group are often referred to as peptides. And examples, we only see this one in amino acids as one of the side chains in amino acids. But those are going to be the 8 functional groups that we want you to know in Bio 180. A couple of things as we go through this. I want to talk maybe about just a couple of patterns in the functional groups that I see that I'll explain to students that might help you learn them a little bit. So, one of the things that I do in trying to learn these is I separate them into different categories, and I'm going to show you how I separate them. You don't have to do it this way. You could do it another way. You could say, Dr. Christensen, that way is stupid, and that's fine. So I'll just show you how I help remember them. I divide the functional groups, or these 8 ones, into 2 categories. One category I call my simple H category, and then the other category I call my complex O category. And here's why I call them that. 4 of the functional groups are just going to be a primary atom like carbon or oxygen or sulfur or nitrogen that are simply then surrounded by hydrogens. So I really just have to remember those 4 atoms, and then I put in the number of hydrogens needed to fill out their octet. So in the case of carbon, I add 3 hydrogens. Remember, we've got an R group on the front of all of them. Oxygen only needs 1 more bond, so that's my hydroxyl group. Sulfhydryl, 1 more bond, and that's my sulfhydryl group. And then nitrogen, remember, requires 2 more bonds to fill its octet. I add 2 hydrogens, and that's my amino group. So these ones are simple because I just put 1 atom down and fill it with hydrogens. And so that's why I call that category my simple H's. The other 4 functional groups are more complex. Instead of filling with H's, though, I'm going to tend to fill with oxygens, with O's. On this one, I start out, the key is starting out, I draw a carboxyl group. Okay? There's a reason why I start out. All of them are going to kind of be derivations of this same structure. For example, if I want to draw a carbonyl group, I can still draw my carboxyl group, and then I just erase this, put on another R group, and that's my carbonyl group. So I'm just erasing part of the carboxyl. If I want to draw an amide, then I draw a carboxyl group, and then I just erase that oxygen, replace it with a nitrogen, add another hydrogen. But it's still, I'm going off of that same basic structure. The phosphate is, of course, the hardest one, as we've said. So this one, again, I can kind of start out with a carboxyl group. I have to erase the carbon, put in a phosphorus, and add 2 more oxygens, and that's my phosphate. So notice that all of these are more complex functional groups, but they're all based on adding oxygens to carbon or phosphorus. And so that's why I call them my complex O's. Simple H's, complex O's. Okay? So that's just one way that I help remember the structures, at least, of the different functional groups. We will overall, again, some of the best way to understand this is really you're going to have to repeat, get a blank sheet of paper, write out the functional groups, write out the characteristics, write out the properties. And then when you get done, try and fill out as much as you can. Once you get done, take that piece of paper, crinkle it up, throw it away. Pull out a fresh sheet of paper, draw the functional groups down one side, the characteristics or the categories along the top, and start filling it in again. Try and do as much as you can from memory. Once you fill it out as much as you can, then go back, look at your notes, fill everything in, then throw it away, start afresh. Okay? I also like to promote or encourage students as they're learning the functional groups, look for patterns like we did with simple H and complex O. And we started relating or connecting the functional groups to each other, saying, well, this is like this, but a little bit different. It's much easier to remember differences between 2 things, or remember one thing and then similarities and differences, than to remember 2 separate things. So I encourage you to do that. So now that we've learned about functional groups, we are going to move on to the next section, which is talking about how we're going to use those functional groups to carry out some chemical reactions and create a group of molecules known as polymers. Okay, so we just finished talking about functional groups. So now let's move on to our next topic, which is talking about polymers. So what is a polymer? A polymer is anything where you have a series of subunits that are connected in some kind of chain. So this would be an example of a polymer where we've got circular units. And so this, we can call it a subunit, or it's often also referred to as a monomer. In fact, mer comes from the Greek word meaning subunit or part, and then poly means many. So polymer literally means many parts or many subunits, and monomer literally means one part or one subunit. So that's one part, and then we connect them into a chain to make a polymer. So a polymer is a chain of identical or at least similar parts. We're going to learn about molecular polymers, but you could think of things like a train is kind of a type of polymer. You can have all the same kind of railroad car. You might have different kinds of railroad cars, but they all connect into a train polymer. So that's what a polymer is. And we're going to see that there are a lot of important polymers in biology. So here's the name of a few polymers that we'll be talking about over the next day, or next couple days in class. So we have proteins are a type of polymer. We have molecules like DNA, you've probably heard of, and RNA are a type of polymer. There are carbohydrates. Some of you guys may have heard of cellulose before, found in plants, or maybe glycogen, or starch, the type of carbohydrate polymer. So we're going to learn about these polymers, and we're going to learn about how they're put together. A common diagram that I like to use when talking about polymers and how they're made, and we're going to go into some detail here, is the following. So in general, we're going to start out as monomers for some kind of subunit. And there's various types of monomers. And then monomers are converted into polymers by a special kind of chemical reaction called a condensation reaction. And so you have this arrow, monomers going to polymers, and that's called a condensation reaction. Sometimes we try and be consistent with calling it condensation reaction, but you'll also hear it called a dehydration reaction. And in some ways, dehydration might be a little bit more descriptive in that when you dehydrate something, what are you doing? Well, you're pulling water molecules out of it, right? Or you're pulling water out of it. And that's what we're going to do. In condensation reactions, we've got a little side arrow that comes here. It produces a polymer, but it also produces water. So we're going to produce a water molecule. So monomers become polymers. They release in a condensation reaction, and that condensation reaction releases a molecule of water. On the bottom half of our diagram, we can also take polymers and we can convert them back into monomers. That takes another kind of chemical reaction called a hydrolysis reaction. And so hydrolysis, if a condensation reaction formed water, and hydrolysis is the opposite of condensation, you might correctly guess that a hydrolysis reaction requires water, and it does. In fact, hydrolysis comes from two words, hydro, referring to water in Greek, and then lysis, referring to cleavage or breakage. And so hydrolysis literally means splitting or cleaving with water. So the name is actually very descriptive. Now, as we go over through class, or in class over the next couple days, we are going to learn about different monomer types. So we're going to see a monomer in carbohydrates called glucose. And glucose as a monomer will undergo condensation reactions to form glycogen, which is a form of polymer. So this is the polymer, that's the monomer. Another example that we'll go through. Lipids will also, they don't quite form polymers. We'll explain that a little bit when we talk more about lipids. But we can take kind of monomers called fatty acids, and they will undergo a condensation reaction to form fats or phospholipids. We can take amino acids, which is a type of monomer, and amino acids hook together to form a polymer called proteins. And finally, the last group, we can take monomers called nucleotides, and we can convert them into polymers called nucleic acids, with the two nucleic acids that are commonly known are RNA and DNA, and you guys have probably heard about those before. So this is just a general drawing, and these are specific examples that we'll be talking about in class. So again, glucose is the monomer of glycogen. These are kind of color-coded as I've written them up on the board. Fatty acids would be the monomer of fats and phospholipids, and so on. So that we better understand this condensation and hydrolysis reaction, we're going to give you a couple of examples of what this actually looks like in the chemical structures. So maybe let's start out with one of the easiest ones to show, or one of the first ones we'll go over actually, involves glucose. So here is a quick illustration of a glucose molecule. And then here's a second glucose. In fact, I'm not going to show the whole molecule, just the important parts. So they do have hydroxyl groups. That one's important. They do have hydroxyl groups, but I'm going to only show the hydroxyl groups that are important. In fact, even that one's not important. So this is roughly what glucose looks like. We've got a carbon right here on this end, and carbon. So these are two glucose molecules, glucose and glucose. And remember, we want to form a new bond so that we hook these two. They're individual monomers right now. We want to form a new covalent bond between them. So this bond right there, we want to form a new bond so that they're hooked together in a chain. So how do we do that? How does it work? It's going to come back to some of the functional groups that we've learned about. And so what's going to happen is right here on this glucose, we have a hydroxyl group right here. We have to reach back into unit 1 and remember that this oxygen has a partial negative on it because it's in a polar bond. And remember, this carbon, we talked about it earlier, is also not in one, but in two polar bonds. It's got an oxygen connected to it right here, pulling electrons away from it. And it's got an oxygen right here pulling electrons away from it. That means this carbon has a very strong partial positive on it because all the oxygens are pulling its electrons away. Well, this partial negative sees that strong partial positive and is attracted to it because opposites attract. And so if they get in close enough proximity, remember this has a pair of electrons right there, and it will actually attack that carbon. And so we say that this carbon is the target, and this oxygen is the attacker. We actually call call this a nucleophilic attack is the chemical name for it. It has a specific name, and so that's why we call that an attacker. It's, you don't have to worry about what a nucleophile is or electrophile. We're just going to call this oxygen the attacker. What do you need to be an attacker? You need to have a strong electronegative pull or a strong negative partial charge. Negative partial charge. And then in any condensation reaction, so you have one attacker, and then you also have to have a good target. So what do you need to be a target? Well, you need to have a strong partial positive charge. And that almost always happens because you have two or more oxygen bonds. And so we've got two oxygen bonds in that case, and so this carbon now is a good target. It gets attacked, and we end up forming this bond. Now at this point we have a problem, because carbon right here at this point now has 1, 2, 3, 4, 5 bonds. But that breaks the universe. Carbon doesn't have 5 bonds. Carbon can only have 4 bonds. So we've got to fix some things here. What do? We've got to get rid of a bond. And the bond we're going to get rid of is going to be this one right here. Now things are back restored to order. I've got 4 bonds on that carbon. But now we've got a problem over here. Oxygen has 1, 2, 3 bonds. But we've learned oxygen doesn't make 3 bonds. Oxygen makes 2 bonds. So we've got to get rid of a bond over here too. So we're going to get rid of this bond. Well, that doesn't make that hydrogen or that hydroxyl group very happy. They need those bonds. So they are instead going to form a bond together, and that's where we form our water. Remember over here we said when we do a condensation reaction, we form water. Well, there's the water that we form. Okay? It's coming from the loss of that hydroxyl and that hydrogen. They get together, and that forms our water molecule. And now we have this new bond going between those two. And now if we wanted to add another bond, we would just bring in another glucose molecule. A new attacker, a new target, form a new bond, break those, form water, and we would just keep repeating that until we had as long of a polymer or chain as we wanted to. And that's how our polymerization reactions are going to work. So again, this would be called a condensation reaction. It's occurring when one of the functional groups is a good attacker, and you also have a good target somewhere on the molecule. They smash into each other due to mutual attraction. And then we're going to break a couple bonds to form water. Hydrolysis reaction is just going to operate in reverse. We're going to take this water molecule and we're going to reattack that carbon, and then it'll break that bond. So we're just kind of switching off bonds. And that would be a hydrolysis, and then we reform here, reform there, and that would be a hydrolysis reaction. Okay? So that's how our condensation reactions actually occur chemically, or the chemical basis behind it. In each one of our groups, the monomers then have a good attacker group and have a good target group on them. As an example of this, let's try one more example on the board. Here's an amino acid structure. Okay, an amino acid, this is 2 different amino acids. They have kind of a short backbone chain here. We've got an amino group right here, hence the name amino acid. And then we've got a carboxyl group right here. Notice we've got 3 bonds to oxygen, and it's drawn a little bit differently, but that's our carboxyl group. Now, as you look at those structures, can you identify which one of those functional groups might be an attacker and which one might be a good target? Remember, that's what we need. We need a good attacker and we need a good target so that we can form our condensation bond. As you look at those and say, well, this carbon right here has electrons being drawn by that oxygen and that oxygen, which gives it a big partial positive charge. That would be a really good target. And you'd be right. In fact, that's an even better target than on the carbohydrates that we drew earlier. So this is our partial positive target. Nitrogen is a strong negative partial charge. It's more electronegative than the carbons and hydrogens, so it's got a partial charge. It's also got an unbonded pair of electrons to attack that carbon, and so we form a new bond there. Carbon, again, has 1, 2, 3, 4, 5 bonds. That's too many. We've got to break one of them, so we'll break that one. Nitrogen now has 1, 2, 3, 4 bonds. That's too many. So we're going to break one. Let's break that one. That becomes our water molecule, and we now have these two amino acids connected together. We could draw a third amino acid here and connect it in the same way, and that's how we would go about building our protein polymer. Okay? One thing that we'll go over in class as we talk about each one of these groups. This new bond that is formed here by the condensation reaction often has a special name associated with it. In the case of proteins, we call this a peptide bond. So it has a special name. That's the peptide bond connecting those two together. And actually, each one of these groups has a special name. So over here, the glycogen, the bond that holds glucose molecules together, is called a glycosidic bond. In fats and phospholipids, we call it an ester bond. In proteins, we call it, as we wrote up over there, a peptide bond. And in nucleic acids, we would call it this one's the biggest one, a phosphodiester bond. And obviously that's related to the ester bond. We'll tell you how when we actually talk about nucleic acids later on. But these are the specific bond names that hold our monomers into our polymers. So we form special bond names. All right. One more thing before we end this polymer discussion. Let's do some application problems. We have a mixture. Suppose I have some kind of container. And I have a container and I have 1, 2, 3, 4, 5, 6, 7, 8, 9. I have 9 subunits. 9 subunits. If I do a condensation reaction, so we'll give you some kind of scenario that will have a mixture of either monomers or polymers or maybe even both in it. And then we will ask you, if we do condensation reactions, when I write RXN, that's just a shorthand notation for reactions. So if we have 9 subunits and we do a condensation reaction, what is produced? Well, we've got 9 subunits or monomers. Condensation reactions, remember, create polymers. So the most obvious answer is we will have a 9-unit polymer. And that would be correct. But is that all we have? The answer is no, because when we formed this polymer, we also formed water. So how many waters did we produce? Well, the quickest answer students often jump to is, well, 9-unit polymer, we produce 9 waters. But that would be wrong. Each time when we bond 2 monomers together, remember it's from the bond formation that we get our water. So if we bond 2 together, we get 1 water. If we bond 3 together, then we get 2 waters. 4 together would give us 3 waters. And so if you start to look at the pattern here then, the number of waters equals the subunits minus 1, or 1 less than the number of subunits. So we would actually produce 8 waters. Now, if we were to take, let's change the scenario, and instead of having 9 subunits, we had a polymer, 1, 2, 3, 4, 5, 6. We have a 6-unit polymer up there. And now we're going to do not a condensation reaction, but a hydrolysis reaction. So we do hydrolysis reactions. Now, if we ask the same question, what is produced? The correct answer would be, well, 6 monomers. And you might say, well, what about water? How many waters? Well, remember, when we do a hydrolysis reaction, we don't produce any waters. But we do require, that wasn't the question we asked, but if we were asked what is required, well, that would require water. So 6 monomers would be produced. We would need 1 water to split each bond that we broke. So we would require 5 waters to do that hydrolysis. Okay? So you have to keep in mind the number of bonds that are being broken or formed and whether water is being produced or required for whatever reaction. Now, hopefully that explains some of those concepts. We're not going to give you a problem like that. Here's going to be the type of problem that we give you. It will be a combination or a compound problem. It will say something like, a solution contains a 30-unit polymer and 25 monomers. If we perform multiple condensation reactions, and then usually you'll put in parentheses something like to completion. So how many did we perform? As many as we possibly could. What would the end products be? Now, this type of question gets a little bit trickier, and what we're testing for, not only do you know what you're doing, but these underlined areas we can often change. Like instead of writing condensation reaction, I would write hydrolysis reaction. And so you really have to understand what condensation is, what hydrolysis is, and whether waters are required or produced. So, how would you go about answering this one? Well, here we're doing a condensation reaction. So we're building a polymer. The tricky part on this one, so most people would understand again, there's a little bit of a trick to this. They would say, okay, our products, we're going to end up with 30 polymer plus 25 polymer or monomers. We're going to add to it. We're going to have a 55 unit polymer. We're doing condensation reactions, so water is also going to be a product, right? Condensation produces water. So we're going to have water. And then the most common mistake, so this is the mistake, bad, don't do this. The mistake is they look at, from our previous discussions, say, oh, 25 monomers, I got this, N minus 1, so we're going to produce 24 waters. Wrong. Why? Because this polymer up here is also acting as another subunit, if you will. In other words, I've got the big long polymer. When I add my first monomer, so I've added monomer number 1, I got 1 water. I add monomer number 2, I got 2 waters, or my second water. 3 waters. You keep going on this. When I add 25, I'll have 25 waters. In other words, it's still N minus 1. You just have to remember that this is one of the things that you can form bonds to, so that we actually have 26 things in that solution. 26 minus 1 is actually 25 waters that are produced, 55 unit polymer. Okay, so I did that specific example just so you recognize that's the only tricky thing. There are other problems that we could give you that are more basic. Again, we could change this to hydrolysis. That changes the products. We could say instead of what the products would be, we could say what's required. Okay? But you can practice with yourself on multiple problems. It's not meant to trick you. That's why we're telling you about it. We just want to see if you can tell the difference between condensation and dehydrolysis, or in hydrolysis reactions, and then what products or reactants might be used in them. With that, that will close off today's lecture. We will be looking at carbohydrates next time. Good luck. Have fun.